So now that we've looked at the background experiment, what we want to see are how are enzymes, either proteins or polypeptides, made. So think of that whole complex of proteins made up of amino acids or our polypeptide chains made. So, how essentially do we get from DNA, this code, this instructions, to the making actually of the physical protein? And so the intermediate is RNA, ribonucleic acid. So it's the bridge between the DNA and the protein. So we're going to have a DNA strand. We are going to transcribe. And so if you look at that, scribe is writing, trans means across. So we're going to write it into a new language that we call messenger RNA. We will dig into this in more detail, but that rewriting goes from a double strand. Now we're in a single strand RNA that we edit, but that messenger RNA as a single strand can now exit the nucleus. Once it exits the nucleus, it's going to bump into some ribosomes, and we'll learn about a small and a large complex to a ribosome that will essentially kind of Oreo cookie sandwich in the messenger RNA, and then we'll translate from messenger RNA into amino acids, and then we'll have our protein synthesized. And so the basic process, again, we're going to go from instructions of DNA. We are going to transcribe, rewrite DNA into messenger RNA. Again, we're in the nucleus here. We don't want to leave the nucleus. We don't want that DNA getting out, because if that cell needs to go through a mitotic event, a cell division, we can't have the DNA just free floating around. We want to keep it locked away, and we just want to make copies. And that's what RNA is doing for us. And then exiting the nucleus so that it can be translated into proteins, because that's where we're going to find our ribosomes. So, a couple of differences. And for the most part, we're going to focus on transcription in eukaryotic cells. But you have to remember, and this graphic isn't the best, but prokaryotic cells do have circular DNA. They make this look linear, so they're just taking one little piece. But that transcription process is going to go, we're going to go from DNA into a messenger RNA. We don't have a pre-messenger RNA. We're just going straight to messenger RNA. And again, we know that ribosomes are free floating around, and so they can sandwich that messenger RNA. So, since there is no nucleus, the protein synthesis occurs essentially at the exact same time as transcription. So, as you're making this messenger RNA, you're going to be transcribing it as you make it. And we'll study that. We'll get into that in more detail. Over on the eukaryotic cell, we have a truly defined nucleus with a nuclear envelope. And so we're going to have to transcribe into a pre-mRNA. We process the RNA and get it. We're going to edit out with introns and exons. We will link together the final messenger RNA that we are going to send through a nuclear pore out into the cytosol. It'll bump into a ribosome, and then the ribosome will start moving the messenger RNA. We're going to see tRNAs come up, and we start making that polypeptide chain. So we're going to go DNA to RNA to protein. And so weirdly, they call this the central dogma. So this is a huge biology concept. Has nothing to do with dogs, although dogs would go through this process. But central dogma, this big idea of how do you get from a DNA code to actually the usable form of that code, which is the protein and polypeptide. So we're going to watch this quick little video. So that this is the central belief that we're going to go from DNA to RNA to polypeptide. Um, so tomorrow we're going to start with this process of transcription. We will go through each of these phases has like subunits that we can break the steps into.